patient lies dying from blood loss. A doctor, desperate to save them, transfuses blood directly from a healthy donor into the patient's veins. Sometimes the patient recovers almost immediately, color returning to their face within minutes. Other times, that exact same procedure, performed the exact same way, kills the patient in agonizing fashion within the hour. For over 200 years, doctors had absolutely no idea why the same life-saving act could either save a life or end one, seemingly at random. Some physicians concluded blood transfusion was simply too dangerous to ever be reliable. Others kept trying anyway, burying patients they couldn't explain. The answer had been sitting inside every single person's veins the entire time, invisible, waiting for one scientist to notice something nobody else had thought to look for. Think about this. You've probably given blood, received blood, or known someone who has. A car accident, a surgery, a difficult childbirth. Blood transfusions today are so routine, so thoroughly unremarkable, that most people never think about them twice. You fill out a form, answer a few questions, and if you ever need blood yourself, it simply arrives, safely, almost every single time. Why does something so simple, so completely reliable today, have a 200-year history of essentially being a coin flip between saving a life and ending one? Not because the procedure itself was complicated. Transferring blood from one set of veins to another is mechanically simple, something doctors figured out how to physically do centuries before anyone understood why it sometimes went catastrophically wrong. You're probably thinking, easy. Old medicine was primitive. Doctors just didn't know what they were doing yet. That's the obvious answer, and it's not entirely wrong, but it misses the actual mystery completely. Because the doctors performing these transfusions weren't incompetent or careless. Many were skilled, careful physicians using the best techniques available, following every precaution they knew to follow. The problem wasn't sloppy medicine. It was an invisible variable nobody on Earth knew existed yet, hiding inside blood that looked completely identical from one person to the next. To understand what was actually happening, we have to go back centuries, to the earliest recorded attempts at blood transfusion in the 1600s, when physicians first tried transfusing blood from animals, typically sheep, directly into human patients. Some patients seemed to tolerate it. Others suffered violent, sometimes fatal reactions almost immediately. The practice was controversial enough that it was eventually banned in several countries for well over a century, considered too unpredictable and too dangerous to continue. By the early 1800s, physicians shifted toward human-to-human transfusions instead, reasoning that blood from one person should logically be safer for another person than blood from an entirely different species. An English obstetrician began using the technique specifically to save women dying from severe blood loss during childbirth, and here's where the real mystery deepens rather than resolves. Some of his patients recovered beautifully. Others died from the transfusion itself, reacting violently to blood that, by every visible measure available at the time, looked absolutely identical to the blood that had saved someone else the week before. For the rest of the 19th century, this pattern repeated itself in hospitals across the world. No consistent rule emerged. The same donor's blood might save one patient's life and kill the very next person who received it. Physicians had no framework for predicting which transfusions would succeed and which would kill, because nothing about blood's appearance, thickness, or color offered the slightest clue. It looked the same under every microscope available at the time. It behaved identically right up until the moment it didn't. Here's where an Austrian physician named Karl Landsteiner changes the entire story, and the way he found the answer is almost disarmingly simple. He wasn't working on some grand-funded research project chasing transfusion deaths specifically. He was simply curious about a basic question nobody had bothered to investigate properly. What actually happens, at the most basic biological level, when blood from two different people comes into direct contact? In 1901, Landsteiner took blood samples from himself and several colleagues in his laboratory, then began mixing small amounts of blood serum from each person with red blood cells from the others, simply to see what would happen when different people's blood came into contact under controlled conditions. In some combinations, nothing unusual occurred. The blood mixed smoothly. In other combinations, the red blood cells visibly clumped together, a reaction scientists call agglutination, forming clusters that would have been catastrophic if that same reaction had happened inside a patient's actual bloodstream. Landsteiner realized he was looking at exactly the mystery that had confused doctors for 2 centuries, shrunk down to a test tube where he could finally study it directly. He identified that human blood came in distinct types, which he labeled A, B, and O, based on which combinations caused clumping and which didn't. A fourth type, AB, was identified shortly afterward by researchers building on his work. Blood from a person with one type contained specific markers, now called antigens, sitting on the surface of their red blood cells, and specific antibodies floating in their plasma that would attack any blood cells carrying a different marker they didn't recognize as their own. Suddenly, 2 centuries of unpredictable, seemingly random transfusion deaths made perfect, structured sense. It was never random chance deciding who lived and who died on the operating table. It was a precise biological reaction, invisible under any microscope of the era, playing out silently every single time blood from 2 incompatible people met inside a patient's veins. When incompatible blood types were mixed, the recipient's own immune system attacked the donated red blood cells as foreign invaders, causing them to clump and eventually rupture, releasing their contents directly into the bloodstream in a way that could shut down kidneys and kill a patient within hours. When compatible types were used, none of that happened at all, and the transfusion simply worked exactly as intended. Landsteiner's discovery meant doctors could finally test blood before a transfusion, instead of simply hoping for the best and burying the patients who happened to draw the wrong combination. Simple typing tests, using the exact same agglutination reaction Landsteiner had first observed in his lab, let physicians confirm compatibility in advance for the first time in the entire history of the practice. The coin flip that had defined blood transfusion for 200 years finally had a way to be checked before anyone's life depended on the outcome. The story didn't end there. Blood typing alone solved the compatibility problem, but doctors still couldn't store donated blood for any meaningful length of time, meaning transfusions could only happen directly from a living donor to a patient in the same room at the same moment. During the First World War, physicians developed effective anticoagulants that let donated blood be stored safely for days rather than minutes, finally making it possible to collect blood in advance and transport it to wherever it was desperately needed, rather than requiring a compatible donor to physically be present at the exact moment of a crisis. Blood banks, and the entire donation system that lets you walk into a clinic today and give blood for a stranger you'll never meet, grew directly out of that wartime necessity. Landsteiner kept working for decades afterward, later co-discovering an additional blood factor called ARSH in 1940, which explained a separate set of transfusion complications and pregnancy complications that blood typing alone hadn't fully accounted for. By the time of his death in 1943, the practice of blood transfusion had transformed from a life-threatening gamble into one of modern medicine's most routine, most trusted procedures, precisely because one scientist had finally asked the right question about something everyone else had simply assumed was unknowable. So what if Landsteiner had never run those simple mixing experiments in his lab that year? Blood transfusion might have remained a last resort gamble for decades longer, reserved only for the most desperate cases where the risk of death from blood loss clearly outweighed the risk of a fatal reaction. Surgery as we know it today, entire categories of operations that routinely require transfused blood to succeed, might have developed far more slowly and cautiously, waiting for someone else to eventually stumble onto the same discovery Landsteiner made almost by simple, patient curiosity. World War battlefields, where blood loss remains one of the most immediate causes of preventable death, might have continued losing soldiers to a solvable problem for years longer than they actually did, simply because nobody had yet asked the right question in a quiet laboratory far from the front lines. Remember the last time you or someone you love received blood without a second thought about whether it would work? For 200 years, that same simple act was a genuine gamble with someone's life, decided by an invisible biological marker nobody could see, test for, or even imagine existed. It took one scientist mixing blood samples in test tubes to turn that gamble into one of the safest, most routine procedures in all of modern medicine. So next time you or someone you know needs blood, and it simply works, remember what's actually happening. You're not just receiving a transfusion. You're benefiting from over a century of a mystery finally solved. An invisible marker in your own blood that once decided, completely silently, whether a simple medical procedure would save a life or end one. Millions of transfusions happen safely today, without a second thought. Now you know why.